Let's start with a little bit of an orientation lecture. So Christine is going to introduce the concept of the scientific method. What is science? How we approach science, how we design a really good experiment, what kinds of things we need to think about when designing our experiments. What are the potential pitfalls and drawbacks in designing experiments, and statistics, and communicating our results and writing up our results, and all the many things that go into being a good scientist, which we hope, regardless of what career we end up in, we hope on some level or another, we are good scientists. So Christine, the floor is yours.
All right.
The Zoom is yours.
Okay, let me just share my screen. Okay. Okay, as Norman said, I just want to talk a little bit about what science is, what's the point of even doing experiments, and what we do after that to incorporate the results into like the greater scientific body of knowledge and public health in general, because we are doing public health here. We really, in this class, we want to teach you, of course, like laboratory skills, but we also want you to come out of Mailman prepared to be like good scientists, to like critically interpret and add value to the scientific community and public health community at large. So we're just going to kind of do a little brief overview of science and like the concepts behind it, experimental design. And we'll have a breakout session where you guys will talk about like your own experiments, and then we'll also have another break sort of in between. We're going to talk about science and scientific approaches. I'll go a little bit into the actual doing the experiments part, a brief overview of lab notebooks, experimental design concepts, so things to keep in mind to really keep it top of mind while you're designing your experiments, because there's really no point in doing it if it's not designed well. It's just sort of a waste of time and resources. And one of the things you start with is like a really explicit question about what the actual purpose is of the experiment. So we'll talk about that and just different design methods, significance analysis, and other considerations when you're analyzing your experiments. After that, that's where we'll take a little break, and we'll talk about communicating our findings with scientific papers, peer review, quality control, a little bit about publication bias, and finally, how to take the findings from our papers, from science, and apply them into reality in a larger public health context. So what is science? So here's thinking of doing a little word cloud. So you might know Pull Everywhere. So you can just you can text Climbing Group 863 to 37607 and just text your response. Or you can go to pullev.com/climbing group 863. It's up there. So just whatever science, what you think of when you think of science, what science means to you, just explore that. Yeah, innovation, definitely. Yeah. Discovery. Oh, nuance. I like that. Yeah, it's interesting. Research. Indeed, brains. Only if it's spelled with a Z, though. Progress. Research. Oh, learning. Ooh, humility. Cool. Money. Time. Data. Frustration. Definitely frustrating sometimes. Yeah, I'm seeing a lot of like collective, community, collaboration. Yeah, that's definitely knowledge. Experiment. Became big. Definitely experiments are necessary in science. That is right on cue. Brain, brains with a Z has been emphasized. Dexter's. Yeah, I love it. Science. Yeah, for sure, Bill Nye. Oh, the stress popped up. Yeah, it is. It can be pretty frustrating and stressful if you're doing it correctly and not sort of landing at whatever result you kind of predict. That's sort of part of the gig. We really like brains with a Z here. Yeah. Yeah, I love these. Yeah, so
Excellent.
excellent. Thank you guys. Yeah, I see a lot of like sort of themes, but also it's like kind of there's so many things that science can mean to people, right? Because it is kind of like, I mean, it's such a broad topic as much as I claim to just be all about science. What does that mean? It means all of this. Does anybody want to like say any, like have any particular thoughts either way about what science means to them?
One thing that's kind of hard to put in one word is, or I guess one word, scrutiny, because I feel like a lot of people
Mhm.
don't believe science, even though when it's proven,
Yeah. Yeah.
getting people to believe it is hard.
Mhm.
Definitely. Yeah. Especially more recently.
Yeah,
Um,
absolutely. I don't know if
Yeah,
anyone
just
saw
a-
the poll that was recently done that asked people what are the top 3 places that they get their facts about the pandemic from? And science or scientists or scientific groups were not in the top 5. It was your church, it was your employer, it was your school, and the CDC and the government and science was way down the list. So we are doing a very poor job of communicating our knowledge.
yeah, it's been tough. That would be the stress and frustration part. I did like humility here.
Very important.
Yeah.
I think Jessica, that's a good point. Part of science is communication, and learning how to communicate
Mhm.
complicated stuff to people who have no experience with what you do. And I think probably we all have encountered that throughout the past 2 years.
Mhm.
Telling people that, yeah, like these vaccines were not made in like a couple of months. Like RNA vaccines have been in the market, like not in the market, but like in research for a long time.
Yeah.
So trying to get people to understand this kind of stuff, um, is stressful, frustrating, and all
Yeah.
the words that are science. So
Yeah. Yeah, it's true. It's become also like so much more. It always was part of science, like a huge part of it, especially get input from everybody. But definitely in the past few years, it's been an extra challenge. So, but also even more important, right? Because now people's health even more than ever is on the line. So, yeah,
Excellent point, Shane.
yeah, very good.
I would add too, maybe, I don't know if it's on there, but I would say like complicated because like maybe if you're in the science field or something to do with medicine or science or anything like that, and you're trying to explain something to someone in terms that anyone can understand, that alone is a challenge.
Mhm. Yeah, that's
yeah,
pretty
it's like,
so hard. Go ahead.
oh, go ahead, Sultana.
Oh, I was just gonna agree with Jessica, and that's why I don't know if this is like a popular opinion, but like I kind of see science as kind of like an art form because like in science you're kind of attempting to like describe the world around you. That's kind of what art does as well.
Mhm.
I agree with that, Santana. Well put.
Yeah.
Yeah, it's a form of art. I will also say that it's going back to the communication part. It's a challenge when you want to explain logic and the other person is coming back with opinion. Um, I've had plenty of conversations where people tell me like this and that, and I'm like, yeah, that's not how things work, that's not part of the scientific method, so good try.
Yeah, it's tough. Anything else that's popping out at anyone? Okay. Yeah, this was all sort of straight on and sort of, I'm just in the history of, just one of the ways where's my mouse? Oh, here it is. Yeah, all of this definitely encompasses science, and right, it in the world, like it wasn't always, people weren't always, well, I guess now people don't always have so much respect for the scientific method as much as we would think, we would hope, but in like the I can't go to my next screen. Oh, there we go. So obviously at different points in time and around the world, there were different times when people were more focused on evidence, but one of those sort of most well-known periods was the scientific revolution, which was, focused on the laws of science, so being discovered by gathering and analyzing data from experiments and observations rather than by using logic-based arguments. This was in about the 16th century, and prior to that, in this sort of area, it was a lot more about like things by like divine decree, or just even like Platonic, like starting from logic and then going forward there, but just like not checking that your axioms were real. And then this sort of obviously revolutionized sort of like a lot of thought processes. He has some good quotes. I just wanted to read them. If a man will begin with certainties, he shall end in doubts. But if he will be content to begin with doubts, he shall end in certainties, which I really liked, because that's the whole thing of keeping an open mind. And then truth can never be reached by just listening to the voice of an authority, which is a big part of how science is about a community. And we do listen to authorities, but like because we respect them in the community and respect their ways of thought. It's not because they're an authority necessarily. And then last one, truth is the daughter of time, not of authority, because as we go forward, we refine our ideas and get closer and closer, hopefully, to the truth. So we know, we've heard of the scientific method, where it keeps going. It's a cycle over and over, where we observe the world, make a hypothesis, design an experiment, carry it out in reality in the real world, and look at our actual results, analyze them, interpret them, and within the larger context of everybody else's results and research and other people's experiments, so that we can get closer and closer to what the truth is, the reality in the world. And then we keep doing it over and over and over, and hopefully we get closer and closer to better models of reality. And again, there's a huge, sometimes you can think of science as like you're in your lab and like this like little ivory tower, and it's not involved with other people, but it's really a community-based endeavor. Everybody talks to each other so that ideas can evolve. That's essential. The feedback from the community, the whole point of communicating, as we were talking about before, communicating to lay people, communicating to other scientists, and really getting more input and more ideas is like the whole point of science. It doesn't work if you do it by yourself. Which is why we have things like papers and conferences and the peer review process, which we'll talk about a little bit more, but that's basically just to a way to have more experts come together, as a community, and make sure that the data everybody's looking at has been like sufficiently vetted. Oops. So this is again, just a little diagram of the process. Ask a question, research, hypothesis. I liked this one because it said, you do your experiment and you see if the procedure is working. If it's not, you troubleshoot the procedure, carefully check the steps. That's okay. But you have to make sure that you report that when you report your in your paper, when you're telling people about that. And then you analyze your data, your results align or don't align with your hypothesis, and the arrows are on both of them. You communicate the results no matter what. And those null results or weird results or whatever can help construct new hypotheses. And as we're doing this, we're refining, we're communicating results, we're refining, we're getting closer and closer to something, to our theories can be closer and closer to reality, we hope. And I just thought we could talk about scientific theory versus scientific fact. And it's sort of like the difficulty of making that jump as scientists and as a society. Does anybody have any like opinions about that or comments?
I think maybe the difference between scientific theory and scientific fact is goes along the same scientific method. You know, hypothesis, something that you have uh, a thought that things could work, an educated guess, versus scientific fact, something that has been proven several times. Um,
Yeah, definitely.
So can someone name a scientific fact and contrast that with a scientific theory?
This may be a bit of a stretch,
Mhm.
but it's a scientific fact that if you have a clean home and you wash your hands and you take care of your hygiene, you are going to be healthy, relatively healthy. And then there's the hygienic theory, I think that's what it's called, where if the more clean your environment are and the more clean you are, the more susceptible you are now to things like allergies.
Okay, so the hygiene hypothesis.
Hypothesis, thank you.
Yep. Okay. Yes, those are, although I might still argue that the former was a little bit more theoretical. There could be exclusions to that rule. But as someone just pointed out, gravity is a scientifically established fact, right? As far as I know. I don't know, Christine, would you argue that that was theory or would you say that was fact?
No, I would definitely, I mean, one of my, I'm happy that you said gravity because, right, if we do like Newtonian mechanics and just F equals ma and like the normal stuff we learn in high school physics, and we look at gravity, it's there. Like I'm comfortable saying that gravity is real. But we have that model, the Newtonian mechanics, but that doesn't work when you're going really fast or if it's really big, right? We have like general relativity, and even if you're just in space, you can't just like the gravitational constant is different. So I think it's interesting to talk about that one because it's a scientific theory that's like we accept it and it like as scientific fact, but we also know that there are limitations to that model, which I think is a really important thing to remember when we're saying a theory versus a fact. Like I'm comfortable saying that like if I do F equals ma on like a rock that I'm throwing, like it's a fact that it will do what I'm telling it to do. But I also can recognize that model, you know, that theory is a little bit, it's, you know, before we had general relativity, we thought that was scientific fact, but it's not. There are limitations. It's actually like a theory that was sort of broadened, which I think is like a fun and a kind of important distinction. So maybe like we have a model, you know, we have like the germ model of disease, and we're like, okay, if there are pathogens, they're going to make us sick. So we're for sure, like if we get rid of them, we'll be fine. And that's a theory that like it seemed pretty clear and it makes sense, right? There's like a pathogen, it makes us sick. But then overall, we started learning more about like the microbiome and like the hygiene hypothesis and, you know, just the scientific fact, which is kind of, it was a theory that was accepted as a fact, kind of like broadened a little bit. Like we are always breaking our models, which I think is important to remember. It doesn't just apply to physics, it applies to public health as well. It's kind of a...
So I'll give you another example. That's a great point, Christine. And I guess I would modify that by saying that it is a fact that gravity works under this set of environmental conditions, not too big and not at the tiny, tiny, tiny scale either, right?
Right.
So somewhere in between it is a fact, but it has limitations. Another fact, which until 20 years ago, we all knew was a fact, was that infectious diseases were caused by bacteria or viruses or something that contains genetic material. You can't be infected with something that doesn't contain genetic material. And along comes Stanley Prusiner and says, no, proteins can make you sick and they're infectious. And everyone laughed at him for 20 years. And he ended up winning the Nobel Prize for completely upending the paradigm and saying there can be proteinaceous infectious particles, prions. And that is now a fact. And maybe there's another fact down the road that we haven't even considered that is going to add and modify onto this basis we now have.
So would the distinction between theory and fact be down to what is observable by the senses? Because I feel like that otherwise it seems like a pretty thin distinction that's really only made by time, and that's still kind of subjective.
I think it is subjective and it is determined by time.
I feel like
Yeah.
like a theory would be like if you go around licking doorknobs, you'll get sick. But what is observable is there are, you know, microbes on the doorknob. So before we have the technology to observe any bacteria or any pathogens, it really would just be a theory, wouldn't it? I mean, you could have a bunch of people licking doorknobs and they get sick, but until you can observe something on it, I feel like it remains a theory.
Observation and testing and validation is absolutely essential to establishing, taking from a general theory to a factual observation. But we need to always keep in the back of our mind that there may be exceptions to that rule, or what we accept as fact may need to be modified in light of new evidence or new fundamental ideas. You know, we all believe that DNA leads to RNA leads to protein. Who knows, in some future world, we'll find some that will be upended in some way. I don't know. I don't think so, but one has to be open to the possibility. One has to be open to new approaches and new ways of looking at data. But at the same time, we have to be skeptical of scientists who present data as totally upending what we see as established fact or established accepted theory, unless we have very strong evidence to the contrary.
I'm thinking with that, I mean, RNA splicing and like, I'm pretty sure that wasn't originally part of the deal, right?
No one even believed it existed.
Yeah, people, and I'm sure when people brought it up, they were like, no, I'm pretty sure that it's DNA and then it's transcribed, you know, and then you get a protein. Sorry, like you're crazy, right?
Exactly. So our history of science is full of these kinds of outliers that turned out to be right and completely added to and upended and changed our perception of the world that we live in. And that is a constant process and will be going on forever, as far as I know. I don't think we're ever going to get to an endpoint.
What do you think is the crazy theory right now going on?
Ooh,
The what? Sorry,
what's a crazy theory going on right now?
That could be true, but people think it's not.
What about the one that life actually originated extraterrestrially and was brought by like a meteorite here? I think it's called the panspermia theory.
Yeah, I think that's less and less thought as crazy and far out. I think that people, I mean, I'm just fascinated by the whole concept that the water that exists on Earth probably all arrived from outer space, from meteorites, from ice on meteorites. And that's how come we have oceans on Earth. That's crazy. Right? Don't know. Yeah, that's a good one. Possible. Yeah.
Yeah.
All right. Great
Alrighty.
discussion. Thank you, everyone.
Yeah, thank
that was
you.
great.
So now I wanted to briefly go over different scientific approaches, but first, this slide's really important. So experimental method, you start with a question and then you think of different ways to answer it. I feel like a lot of times people can sort of get these ideas and it becomes a little bit nebulous because maybe we can do this and this thing exists, but unless you know, if you're actually going to go forward and design an experiment and do it, you really need to start and with a clear question and then start thinking of ways to answer it for it to be to stop yourself from like falling into little traps. And I'm going to talk about like a bunch of different scientific approaches. And I was thinking we could go with like some sort of like association or question or public health question that we have. We like, for example, like do cockroaches cause asthma? We could do that. We could do a different question if anybody had any ideas, something like that. Any favorite questions?
For example, Jessica mentioned the hygiene hypothesis or anything else, something that you came across that, oh, I know we talked about that in the core, or we talked about that in class, or I read about that in an article.
Okay, do you want to just do the hygiene hypothesis then? Maybe cockroaches? Anybody have any preferences?
Let me have one thing. I'm trying to think how I'm gonna put it together in my head.
Huh.
Perhaps exposure to soil helminths and psychological development in kids. So we know that ascaris and chucras and hookworms, these parasites, they tend to absorb all the nutrients that kids need during the critical period. And most of them end up stunned. I wonder how that affects in the long term psychological development, for instance.
Great, yeah. So how would we phrase that as like a question, like to start with our studies? Just like a really simplified version.
The psychological effects of soil transmitted helminths in children.
Yeah. Okay, let's do that.
I'm watching everyone's suggestions here. These are all great ideas.
Oh, in the chat. Oh my God, 17 in the chat. Ah.
So all great ideas. And this is just a small part of the lecture. These are, I mean, we're not going to
Yeah,
spend
this
a lot
isn't a
of,
huge deal. Yeah.
Thank you. These are all great ideas. I like the COVID idea. And A, you had a great idea about subways. They're all excellent.
Aliens. Sorry, I'm just, just reading the whole chat right now. Okay, what do you guys want to could COVID? Oh, wow. So what do you think, Svetlana? Do you want to do the exposure to development? Okay, yeah, okay, let's do that. So just wanted to do a brief overview of of different scientific approaches. Right, we have observational studies where we don't interfere in any way. We have the qualitative methods that like case studies, focus groups, ethnography studies, historical analyses. We have the more quantitative epi methods like a cross-sectional little snapshot or some over time like case control or cohort studies. Can you guys think of any of these that would work for our current question? Any kind of ideas about what to do that would fall under this tree?
Can you repeat or put in the chat what the question is that we're doing? I kind of heard some of it, but I didn't catch
Sure.
everything.
So Richard, I hope I'm not paraphrasing too badly, but that worm infections, helminths, in young children, because they can scarf needed nutrients from the digestive system of children, lead to developmental and or behavioral or cognitive deficiencies that appear on behavioral or cognitive tests later in life. Is that accurate?
Yep, that's it.
I think since there's such a strong time lag from the exposure to the outcome, you probably would want to do a case control study of some kind, just for feasibility purposes. But we could do it I don't know how you would get childhood hookworm levels, but if you had the outcome today of certain cognitive dysfunctions, you could look back and see if as a child, if you had higher levels or not.
Mm-hmm. Yeah, definitely. I'm gonna just write it down and write it in the chat. Okay, I'm gonna go do hookworm infections that, what do we say? Steal nutrients. I'm go with that, whatever. Steal nutrients in young children lead to behavioral or cognitive deficiencies It's gonna be some typos later in life. Does everybody like that question? Does it sound like a good one to answer? Okay. There we go. Now we can be. Yeah, that's definitely a great way to go because it's like, yeah, the data is like pretty like more available. Yeah, I like it. Anything else like that you can do just like strictly observational?
So pick one of these little boxes and say, what could I do? What kind of data could I grab that would answer one of these questions? Case studies, historical, cohort, whatever.
You can
We
do,
can
sorry.
go ahead, Richard. Oh,
Oh, all right. I think you can pick anything actually. Like you can do a cohort study to evaluate cognitive development across years and how they end up, if they had an early infection. You definitely need to collect ethnographic data and see the conditions in which the kids live in. If their nutrition is adequate or not. No, I think that helps.
Yeah, I mean, definitely like everything helps, right? We in science, like we're aggregating all the data. The more varied the methods, the better, because we get all these things to consider from all these sources and methods and techniques to see if we have like some sort of question and we're trying to answer it. The more sort of like evidence that we have from different approaches, the more like likely I am to believe that our answer is close to the truth. Yeah, so there's all sorts of things that you can do.
I think after we see a correlation between the independent and dependent variables, we should do an RCT on some animals to establish causation, and then we can do future experiments after that.
Definitely. And thank you, because that's the next slide. So we can do things with observational data. We could also have experimental study designs, which is the difference is that we're not just observing and gathering data and analyzing it, we're actually stepping in and making a change and looking at the difference that occurs after that change. So yeah, definitely like RCTs are sort of like the go-to thought when we're thinking about like an experiment. And right, so we can do studies like that. We also can do studies in the lab, like in vitro, which means like maybe we have like a bunch of like a cell culture that we'll expose to something and look at that. In vivo would be like an animal study. In silico is like a model in a computer. There's all sorts of different things that we can do where it's like we're making a change and seeing the results. So it's better for causality. Any thoughts about? So that's definitely, we could do like some sort of randomized experiment for our question. Anything else that pops out here?
So on the non-laboratory based side, with human beings, how could you design a study to help add data to the concept that worm infections in early childhood lead to longer term cognitive or behavioral changes in children, and perhaps even an interventional based study? So if we have a population in a geographic area that is susceptible to worm-based infections, and say we have a fairly large population of children in some geographical area, and you want to follow them for a long period of time, and you want to divide them into an interventional and a non-interventional group, what kinds of interventions could you propose that might help validate your hypothesis that the worms are the origin for their future cognitive or behavioral change? And it's not due to some other factor in that environment or in that geography or in their culture.
We can relocate some residents to another area and see the benefits. If the benefits are clear, we can maybe establish causation.
I guess
Good.
you'd have to look into their backgrounds and see what children might be exposed to certain factors, like if they play in certain fields or if they have certain pets or something, and maybe determine whether that is where they may have gotten it, I guess.
Any other thoughts?
I mean, okay, sorry. So I think if, like, theoretically, let's say we have like money, whatever, all the things. If you're in a population where exposure to hookworms is fairly ubiquitous, you could have an intervention, because I'm not super familiar with hookworms, but I believe they have to do with inadequate water and sanitation, that kind of stuff. And so you could have an intervention in which you introduce like maybe filters into the homes, or more sanitary, like you kind of have an intervention to increase sanitation. And I think there are potential other confounders related to that, but I think that's one way you could design an intervention that would potentially make it so that you have controls that are not exposed to hookworm infection early in life.
Mhm.
Yep.
Right, fairly simple intervention too, right? Okay.
All right. Just quickly before we move forward, I think it's really important to emphasize exploratory studies. So an exploratory study is, I mean, you've heard of it, right? So we have an idea, and we do a lot of tests to see if we find any associations. It can be assay based, like for example, we see people who have COVID, and we run a lot of different assays on their blood and see if any of them provide association. We can run a model. We can find like big data we can get from existing data sets, like really large data sets, and see if there are any correlations that show up, or we can generate our own data sets. And these experiments are great. They're really important. We're doing them. But I think it's really important to remember that I just called it an experiment, see? Studies that it's not an experiment. We didn't look for causality. We're finding correlation. We didn't change anything. We're generating data, or we're taking data and we're looking at it. So it's a jumping off spot for other people to do experiments to further explore that idea. And especially nowadays with like big data and everything, they're becoming more and more common. And it's like, I think it's just really important to remember that they're exploratory, which is great. It's just a distinct category. So we can have like existing data sets, like on the internet, you can just like dredge like all of Facebook or something. And we can like generate our own data sets. Can anybody think of maybe like a data set that would be like a big data thing that we would generate ourselves? I would be in that category. So nowadays we have, we've been developing all of these machines that can extract all of this data from samples. that, and it's a machine, so it just like spits out thousands, millions of data points, all sorts of them. Is there, can anybody just like on the news or whatever, like something that they remember that's kind of interesting? Like, what do we use like stats on big data for?
Well, definitely more recently COVID deaths, right? We use statistics to find out what the death rate is of COVID in different areas. Is that what you mean?
I'm thinking, I'm more thinking like high throughput machines, right? 'Cause-
Oh, okay.
yeah, it's, I was just kind of guiding it there. Right, we have like, for example, like RNA sequencing data, we can like sequence genomes and we can just look at all this data and look at all these associations and get some really good jumping off spots. We can do genome-wide association studies, all sorts of things. And, nowadays they're more and more prevalent and they're great. We just need to make sure we take the data and then use it for further hypotheses before like jumping on it. Yeah, that's just where I was going. So there's all sorts of stuff that you can do in silico. You can make a model and just like run stuff, anything like that. So anybody have any thoughts about that? Where are we?
Mhm.
Oh, we use automation and sequencing in COVID testing. What is widely? Yep, definitely. Yeah. Yeah, nowadays it's so exciting, like science can move so quickly because we get all this data and get all these ideas. We should make sure we explore them. Yeah. Okay, now we're going to have a breakout session and talk. We're going to break you up into four groups, and two are going to be group A, two are going to be group B. We're going to talk about ways that we could answer an environmental health question, like really just like any way. We're going to send you this in an email, and I just have it on the screen. Big data would be definitely helpful with social analyses. I mean, that's a big part of like internet, like Facebook stuff and like, social networks and clustering different behaviors and stuff. They use that in some places for like credit, which is like scary. But yeah, that's definitely a huge thing. It's like an exciting blossoming field. Yeah. Mhm. Or even like when people like chat online, stuff like that.
So I'm going to make 4 groups randomly, breakout groups. The first 2 groups are going to do scenario A, and the second 2 groups are going to do scenario B. And Christine will tell you what we want you to do with these 2 hypotheses or these 2 scenarios.
So just like we were talking about, coming up with a really specific question, and then a way to test it where we'll come up with, we're going to operationalize our variables. So we'll have a specific question and then talk about how we're going to measure it, in our actual experiment. So operationalization of variables, we'll talk about it. It's just saying, like, for example, like with the hookworm, it's, if we're in a lab and we're doing it and we're saying, oh, if the hookworm's there, then the nutrients aren't there. And if it's not, they are there. Well, we're operationalizing presence of a hookworm as absence of nutrients in like a cell dish. So we operationalized hookworm as presence or absence of nutrients. There's a tons of stuff that you can do to operationalize your different variables. But so just like ask a question, be clear about your variables, and then like just design a study with like literally any study. We just talked about a couple dozen types of studies. So you got to get a little breathing room and range here. Yeah.
So we'll give you 10 minutes. Christina and I will pop in and out of your various breakout rooms, listen to what you're doing, and then we'll come back and talk about it in 10 minutes. So I'm making 4 breakout rooms right now. And you've all got an email with these 2 scenarios
I think that's everyone, right?
in it.
4 times 4 plus 2. Okay. I guess we'll just go in order, and if you guys want, you can just sort of talk about what you guys talked about, and everyone else in the other groups, feel free to chime in. So go ahead, group one. Oh, just really quickly, I don't know if did everybody read both things or just their section? Go like this for both. Okay, a couple of pieces. Okay, I'm just going to read over both sections so that group 2 knows what group 3 and 4 know what group 1 are talking about. So scenario a large coal-filled fire plant in the south stores its non-combustible residue after burning in large ponds next to the power plant. After a heavy rainstorm, the dam holding back this pond containing thousands of gallons of this material failed, releasing fly ash residue downstream for miles, some of which becomes inhalable airborne particulate. About a decade after a years-long cleanup involving hundreds of workers, a variety of adverse health effects are reported. We hypothesize that some, but not all, of these health problems are related to the fly ash spill. How can we design experiments to assess correlation and/or causation? Okay, take it away, group 1.
Group 1.
I can start. I don't know if anybody wants to chime in
Yeah,
first.
I'll jump in. But go ahead.
So preface, we don't know what to do, but we have some ideas. So we were thinking about, well, Yvonne made a good point of doing like a case control study. And what we would do essentially is have the workers that were exposed during the cleanup and kind of compare health outcomes to workers that weren't exposed during the cleanup. There was like a bunch of confounders that I think Yvonne pointed out too, that workers who weren't exposed to the cleanup could be exposed to other workplace exposures. I don't know if Yvonne wants to talk about it.
No. I have nothing to add. You can go.
Okay, great.
Yeah.
So Yvonne was talking about maybe having residents of the nearby area kind of being our controls, but then the confounding to that would be maybe the residents had the same exposure during the cleanup. So there was a lot of back and forth.
Yeah. Yeah, that's definitely a challenge, right? With real world studies is the whole confounders problem. It's definitely, the world is a really complicated place, and that's the point of doing it in a lab, but then you have to simplify it, and it's just like, oh, that's why we do so many many studies. Yeah. Does anybody have any thoughts about that?
Also, sorry, we just kind of added thing within the scenario because like we took occupational health. So we're under the assumption that the workers have like badges that kind of take in samples of like air periodically. Can we assume that in this scenario? Because that would be very helpful with data analysis.
Exposure assessment, right? Really important. By the way, this is loosely based on a real scenario, and the answer is no, they didn't.
We're screwed. We're screwed.
Yeah, so we talked about like if we want to test the air, that we would need to know what specific particulate we're looking for, because we cannot test for everything. So we will need to look at the workers and what disease they have to have a better idea of what particulates we can look for in the air that are related to the disease.
Yeah, that's really important. You're right, operationalizing your variables. Like I'm going to keep saying it, but determining like what are you even looking for, and is it even a fair measure, right? Yeah. So cool. Is that pretty much it, or do you have more? That's it. Okay.
Yeah,
all right.
I think yeah.
Okay. How about group 2?
Okay, so I guess I can start. So we kind of were thinking similar things. So like potentially an occupational case control study, where you're looking at those that are exposed to high levels, so those that are actively involved in the cleanup versus those that maybe are in the same industry or same company, but maybe like our administrative staff who would not be exposed to any of this, and look at the different health outcomes. But we were unsure what those health outcomes would be, so that was difficult. But and then in order to operationalize your exposure or like assess the exposure, we also thought about potentially looking at biomarkers. So if you're exposed to higher levels of this, it might be able to be found in the blood or urine, saliva, all the things that we talk about. And so that's one way that you could assess the actual exposure levels. Or you could just estimate. I mean, if you could kind of look at the cumulative, you make like an estimation of the cumulative exposure that someone has if they've worked in cleanup for 3 years. So that's what we were kind of thinking about. And anyone else want to add anything?
So Josh, do you think that administrative workers and cleanup workers are equivalent, except for the fact that one was?
Certainly not. And it's obviously a very good question. I think we were just trying to think of getting around kind of the, what's the word I'm looking for? The healthy worker effect that you see in occupational studies. And so they would be in the same like industry or like the same company. So you would get around some of the healthy worker effects that you would maybe see if you took someone from the general population instead. So that's what I was thinking about, but obviously there's many other potential reference groups.
Yeah. Awesome. I love how meticulous you were about operationalizing variables. Okay, great. I'm going to read the second one so that the people in the first two groups know what's going on. Scenario B. GIS mapping of asthma incidents in Manhattan reveals a high prevalence of asthma cases in young children living next to the Cross Bronx Expressway, a roadway frequented by large diesel-powered 18-wheel trucks. We hypothesize that the diesel fumes are related to asthma incidents. How can we validate this hypothesis, and what are potential confounders? Group 3.
So I guess I can start us off. So we wanted to look at this from like a couple different viewpoints. So first, we wanted to have an experimental design where we look at an animal model to see what the effects of diesel fumes on like just asthma development are without other confounding variables. And then to look at like an observational type study where we compare the region right next to the Cross Bronx Expressway with another area in New York City with similar demographics that's away from a major highway like that, and then look at kind of like the prevalence of asthma in that. We also talked about doing a air sample, like an air quality sample, to look at what other particulates are in the air that could be confounding variables that could also potentially cause asthma.
Nice. Yeah.
And for the like the animal study that we were looking at doing, we were like going to expose the mice or rats or whatever we use to like diesel exhaust particles, and then look at like allergen specific IgE and like lung inflammation, airway hyperactivity, like to indicate that like diesel directly causes asthma. And yeah, some other things that we talked about were like we'd have to look at like their inside their home as well to see if like they have pets. We'd have to control for all those factors, like for mold, all of those things. So those are just a lot of confounders that we'd have to look at.
Yeah, definitely. Cool. Yeah, so we've got both observational and experimental in there. That's good. Looking around. Anything else? Anybody have any comments? Alrighty, how about group number 4?
Yeah, so we thought we would operationalize our exposure by looking at air quality index. So diesel is composed of so many different contaminants. So We found that this website had all of the air quality factors to test. So carbon monoxide, UFPs, nitrous gases, VOCs, and test that in the air in the neighborhood that these children are living, and compare that to a neighborhood that is not located near a major expressway. And either when we talked about like either looking at prevalence of asthma or looking maybe at asthma severity. So you could possibly like look at, take samples of these individuals, either in the neighborhoods closer or the neighborhoods farther away, and then compare the two, whether it be by taking surveys of the individual's children to see how severe their asthma is, how many attacks they have per day, per week, that sort of thing. And we also thought about like certain confounders, if they, a lot of them like were listed by the previous group. So living with pets, mold, dander, living closer to factories and other potential contaminants of the air nearby. So that's what we thought of. We actually talked about this case, I mean, something very similar in a spatial epidemiology class. And so looking at the roadway as a line and then inverse distance weighting asthma cases and seeing if there are more asthma cases located closer to the roadway itself. That's just like another observational way to go about it.
Yeah. Awesome. Cool. Great. That was great, guys. Thank you. Any last words, or should I keep going? Okay, I'm gonna share my screen. Okay, so next we're going to talk about doing the actually doing the experiments, right? We've talked about designing it, operationalizing the variables, asking a question, and all the different things you do beforehand. Now I want to talk about what we're doing while we're doing the studies. And I'm going to just start off with, lab notebooks. So why does your lab notebook, hold on, let me move this. Oh, sorry. Okay. Why does your lab notebook matter? Who cares? It's just for you, which it is just for you mostly. A lot of people have this, sometimes people have like this idea that they're like, you have to have your lab notebook so pristine and perfect that if you get hit by a car or you just hate science, somebody can like follow it exactly and do your exact experiment. And it has to be like this publishable thing. But I mean, it is for you. You're not about to publish your lab notebook, but it is really important for it to be detailed enough that it's enables other people to replicate your experiment. And so just a quick review of replicability. It's if someone else, like a different group, or you do it again on a different population, a different group does your exact same experiment, follows your steps to see if they get the same result, which is obviously really important in science. There's a little bit, I don't know if you've heard of the replicability and reproducibility crisis, but a lot of experiments that are given a lot of weight, actually, people have tried to replicate them and don't find the same association. So it's really important to enable people to do that, to make sure we don't run around assuming certain associations are robust when they might not even be there. So that's really important. And it also just reminds yourself to what you did, like your memory isn't perfect, and it clarifies. So when you're writing things up and when you're talking to other scientists, you know what you did, what you were thinking, somewhere to write your thoughts so that you don't lose it. I mean, you're going to spend quite a bit of time thinking about your experiment. It's operationalizing your variables, why you're doing each step. And it's just worthwhile to write it down and reliably keep track of your data. Garbage in, garbage out. So if there's like one thing you take away, well, two things would be operationalizing your variables. And the second thing would be garbage in, garbage out, which is if you have an experiment that's not going to produce reliable or good results, like there's no point in doing it. So if you have your data's messed up, even if you have an amazing experiment, you just wasted a bunch of time and resources and potentially animals if it's an animal experiment. And even if your data is great, if your design is flawed, it's still just like, it's just a waste of time. So garbage in, garbage out is, I'm going to mention that quite a few times. I'm partial to paper because electronic data can just disappear sometimes. It's a little fallible, and then you can find what you were actually doing on paper. But you can also do digital lab notebooks. It's up to you. Anyway, some pro tips include a summary. I'm going to send you guys the slides. A summary and rationale of why you're doing what you're doing, the steps, why you did each one is important because you're thinking about it anyway. Your calculations so that you make sure, for example, for your dilutions and concentrations, that you did it correctly, you have a record of it. And, if you messed it up, like it might still be okay, but you need to know that, what happened. Definitely usually write your sample numbers so you know which sample this applies to, if that's applicable, and your results or the address for them. So if it's like some readout on a computer, just write down, the folder or something so you know where to find your results. Put the date. I think it's important too to cross out mistakes and changes with a line to explain in the margin instead of like scribbling it all out so you can't read it, which I know is tempting because you don't think it's true anymore, but you wrote it for a reason. Or even if you just made a mistake, it's, later, that way you can still read it just in case you want to like, you know what the mistake was. Again, it needs to enable replicability. And my personal opinion is that you should write down your thoughts, your development process, your decisions, and your operationalization justifications, because they're really important. You're going to need them when you're writing up your study. And when you go forward a decade, if you want to look at your stuff, like it's just really important step by step. You might as well, you're thinking about it anyway. And I'm a big proponent of that. So just, even if you're not doing your experiment right then, you're allowed to use your lab notebook. Just writing down your thought processes. If you're talking to people or you're just thinking about what you're doing, your justifications, a thought process, some whole like idea that you were thinking through, and then you discard it because you realize, oh wait, never mind, like this whole grand idea I had actually like doesn't make sense, and then you discard it. Or I don't have the money to do that experiment, so you discard it, but at least you have a record, just in case you think, oh wait, what was that thing I was thinking of a while ago, and then you can just check and just write down why you're doing things, why you're doing whatever you're doing. So now I just want to talk about just sort of concepts about your experimental design. I'm going to talk about my main point other than operationalization, which is garbage in, garbage out. So really design your experiment well, or just don't even bother. Don't perform it at all. Just, it's worth putting in the effort and the thought at the beginning so that you don't just waste your time, and you get results and you publish them and they actually contribute to science, and you utilizes your resources well, and it contributes to society. So Oscar the Grouch, he says, design your experiment well or don't perform it at all. That's what I decided. I really like this this image that describes what I was talking about before, which was that it's both your data and your model that have to be good, so that your output, the results of your experiment are good. So if your data is trash, like it's contaminated, or it was left out too long, or just something's wrong with your data, even if your experiment was beautiful, the result is still trash. If your data was perfect, there's no contamination or anything, but your model didn't make any sense, the result is still trash. So just always make sure that your model makes sense, your data is good, so that your results matter. It's very important. All right, so when you're designing your experiment, as I was saying before, you just have to think about what the purpose is instead of, maybe just jumping in and doing a bunch of random stuff. Just be mindful and think about a question, what the purpose is of your experiment. I put this slide again because it's really important. You start with your question and then you answer it in different ways, like all of the things that we just discussed. We had all these answers to these two questions. Operationalization is important. I've mentioned this 1,000 times. Experiments aren't in the real world. We need to really justify what we're using as a proxy, what it means and why. Write that down in your book, write that down in your paper, and have it clear in your mind. It's important for reproducibility and interpretability, and just remember to do that. Generalization is one, making sure you understand how your results will apply to the real world. If you're doing, if you're looking at like air, certain toxins in the air and you're putting them on rats, like how does that apply to the real world? Well, like maybe you'd have to find out where those toxins are in the air or whatever it is. Like there are 1,000 things, but again, instead of just diving in and looking at something, be really purposeful about what this application will be when you're thinking about it. And because these approximations are just that. Another thing that we use the word generalization for is can the findings, something to think about is if the findings you have, if they can be used for other questions. Like if you have a finding of something that successfully destroys COVID, will it also successfully destroy influenza? Will it destroy bacteria? Will it, like, if this, sort of sanitation method, does it apply to other things? Populations, if you do something in a certain population in New York, does that generalize to the whole country? Does it generalize to the world? Just things to be mindful about is the generalization, and whether it does, whether it doesn't, and if you do think it does, just remember your caveats. And again, just think about it during every step. There may be certain steps where it's kind of irrelevant, but there may be 1 or 2 steps where it's kind of a stopgap that you have to consider. Again, what's the purpose of the experiment, regardless of what the findings are? What will that bring to the field? And a replication of other experience absolutely counts. That contributes to the field because it's replication, verifying or not verifying the results of another experiment. Same with reproducing other findings, also counts. So reproduction is when you have different experimental methods arriving at the same conclusion. So you may have operationalized your variables differently, you may have performed it on a different population, but you ended up with the same conclusion, that there was or there wasn't some sort of association or causation. So the main thing here to enable reproducibility is the careful operationalization, being intentional when you write, and using appropriate statistics. Of course, if your statistics aren't working, your study will probably not be reproducible because it has nothing to do with the truth. And then replicate is when, again, different scientists do the same thing, get the same result. So take good notes, do a good method section, and do your stats well. These are just so important, especially right now in science, to make sure that things are reproducible, things are replicable, and to do those, to be open to everything you do. Doesn't have to be like this novel, groundbreaking thing. That's actually a problem, because if everything's groundbreaking, then we aren't checking and making sure that the things that we think are groundbreaking are actually, potentially supported by evidence. They might not be. It might be like a one-off study with either a small sample, the sample size is small, there was some confounder, or just by chance, it happened to have like a result that doesn't really apply to the world. So just want to talk a little bit about that more, about the methods, the significance, like looking at significance of results and other considerations when you're analyzing your experiments. But yeah, really quickly. So for certain considerations, we have like replicability and we have reliability. And I just wanted to go back here. Just wanted to talk about, has anybody heard about that crisis or have any like examples of it? Does anyone want to comment on it at all? Because I know it's a hot topic.
I thought of it earlier when we brought up like what was science, like what's fact and what's a hypothesis. And I was like, how do you know that like anything's really a fact if like nothing can be reproduced? Like, I guess general trends can be, but I guess if you're looking for like a specific value, then I don't know if you'll ever find it.
Totally. And I mean, even trends are sometimes they aren't being reproduced or and replicated. The exact same experiment, people aren't getting the same result. It's a huge problem. Because it's not how science, you're supposed to do things over and over, right? Yeah, definitely. It's relevant there.
Give you a very famous example of a failure to replicate. 2 eminent groups were trying to reproduce a mouse experiment, and both groups knew what they were doing. They had almost identical procedures. They did the same experiments and they got very different results. and they scratched their head and they scratched their head and they tried to compare notes. They couldn't figure out why they were getting different results. It turned out that the air conditioning system in one of the laboratories was emitting a high-pitched sound, which only the animals could hear. And therefore the animals were behaving differently and responding differently to the environmental stress than the animals in the other laboratory. And that ultimately turned out to be the difference. So sometimes you're doing everything right and you can get the same result. And then you have to go through your notes and go through your methods and figure out why isn't it working? So to answer that, what would you do if you were one of these eminent scientists in the two different labs and you had a population of mice in your lab and a population of mice in the other lab? They were both a particular strain. They were both treated in the same way. How could you verify that it actually was something in my lab rather than something I was doing differently that was accounting for the variability in the result? And this is something that happens sometimes, or with a cell line, or with a virus, or with a compound you're creating in the laboratory. What's another way of verifying replication? What if I send my mice on a field trip? Right? We exchange mice. We exchange cell cultures. We do the same experiments with everyone, with each other's samples. We should get the same results. If we don't, there's something operationally different that we haven't accounted for, some confounder that's we haven't thought about.
Yeah.
So one of the, Christine, you put in the notes, right, in the files, that paper in Science or Nature about the replication crisis and the reproduce. Yeah. It's worth reading. It's really quite disturbing how many studies and really top-notch publications have not been able to be replicated.
Yeah. Luckily, people are a little bit more aware of it now, so they're trying, they're trying a little bit more to reproduce and replicate studies and take them at face value, because it has become a hot button topic that things aren't reproducible or replicable. So at least now, at least people are trying to do that. But yeah, it's disconcerting, and it also doesn't really help with the public perception of science being, it's as tenuous as it is. Yeah. So, chat. Yes, you can absolutely publish papers on replication. I'll talk about that. So it's, unfortunately, if it's not like this cool, exciting thing, it's harder to publish in like a fancier journal. But again, nowadays, because of this crisis, it's people are more open to it. It might be in a lower tier journal, but absolutely, you should, and people want you to publish papers on replication. And if you replicate the experiment and you find the same thing, that's super valuable. It's people get a little too obsessed with like something, pushing the envelope and doing things sort of new, which is important and really cool. But we also need to make sure that the facts and the things that we think are facts and the ideas that we're using to make our new hypotheses are merit that weight by replicating, just replicating famous experiments. So there's some labs nowadays actually that's all they do. Which I'm really happy about. They replicate experience. It's upsetting because I think like more than half of them were not of these like quite famous and influential. There's this paper on it. I'll post it. These quite influential experiences, especially in like certain fields. Is that study on Canvas? I put it in the drive, but I will put it on Canvas after class. Yeah, they're pretty cool. I'll put the study I was just referencing. It's like an academic paper, and then there's an article that's already there that I'll put in the files tab if you guys want to read it. It's really important and interesting stuff. It's going to keep coming up in the presentation. So yeah, so I'll do that after class. I'll put it in the files tab. We can use that. Yeah. Okay, so reproduction and replication is really important. And we need to keep that in mind when we're designing our experiments, and even honestly thinking about which experiments to do, right? But yeah, it's just really important to remember. So just going into things to increase the chance that your results will be reproducible and replicable, just different considerations during your design method and different ways of looking at the significance of your studies and considerations around that. Also, when you're looking at the significance of other studies to design your personal experiment. So we have replicability and reproducibility. There are all sorts of bias that we've learned about. There's experimenter bias, there's population bias. It's always really important to remember to like account for the biases the best you can. And just think of all the biases and mention them. Always think about confounders. You guys did a really good job in that when we like broke up into groups, all the confounders, it was great. Can you guys think of any other like considerations that often sort of pop into your brain when you're designing experiments to make them like more robust?
I was just thinking about safety.
Oh, yeah. Like safety for the people doing the experiment or as a...
Yeah, safety for the people actually performing the experiment, and I guess feasibility.
Yeah, definitely. Oh, feasibility is a good one. Yeah, that's definitely a consideration, just like ethically, right? There are a lot of ethical considerations, safety for the subjects and safety for the scientists that are doing the study. Yeah, that's a great point. Definitely. Thank you. Yeah. Anything else that like jumps out at people? Okay, I'm just going to go through a few of the sort of like classic ones. Sampling, we all know about sample bias. We try the best that we can to do random sampling to find heterogeneous populations if we're doing it in a lab, but sometimes it is what it is and you have to do a convenience sample. But just do your best and describe the rest. Do the best that you can, keep good notes and make sure that you just note what's going on and be aware when you're interpreting the results that, maybe it wasn't as random of a sample as you thought. And if you can get a random sample, it's worth time and resources to do that, to get that random sample. And speaking of that, as you know, like balancing power effects, because if you have like you suspect a really big effect size, you don't need as big of a sample size to have the same amount of power. But if you don't expect like a huge effect, you have to get a lot more people or samples, to have a sufficient amount of power to have, your results really like be somewhat meaningful, but then you have resources to consider. So as you know, it's always just like a give and take. But one of the things that, if it's possible, it's really should be done. You really should do it before the experiment. And it sounds a little crazy sometimes because unless you're running off of somebody else's experiment with like a better sort of idea about the effect size, that makes it easier to calculate the sample size needed for your desired power. But sometimes you don't know what the effect size is going to be. And sometimes you kind of have to guess and it feels weird, but you do the best you can. You note that you decided on the certain sample size because you wanted a certain power. And you thought the effect size would be around a certain amount. People sometimes do a post hoc power analysis as well to say, oh, this is the power of our experiment with the effect size that we found. But that's like a side note. That isn't the main reason to do this. It should be done beforehand to sort of estimate the sufficient sample size needed. There's this really cool program called GPower, and it's like a GUI that you can play around with the sample size and the desired power. Like you change all of the different variables and different ways to look at it, and it gives you like these really cool graphs. So I'd really recommend playing around with that if you feel like it and you have the time. And there's also a really good Towards Data Science article that I will post in the files tab because that's what we are going to do in this class. And again, we always have to be aware of like false discovery rates, type 1 and 2 error. So finding something, saying that evidence supports an association that is probably actually not supported by the majority of evidence, or saying there's no association when there maybe is in reality. And again, we like sensitivity and specificity when we're doing like that's hot topic with COVID, balancing how bad is it if we get if we just say that there's no association versus an association that there really is. Like maybe like a safety study. It's pretty bad for us to say that we don't see any evidence of an association when there is one. So you're going to be a little bit more strict in that respect. Any other examples people can think of? Just like give and take. Okay, I mean, so one thing too, you can think about is for, this is more of a sensitivity and specificity thing, is the seriousness of the disease you're testing for. If you're looking for HIV, you want your test to be super sensitive, because if you get a false positive, I mean, okay, you get another test. But if you get a false negative, it's really bad. Versus other things where you can kind of trade off sensitivity and specificity, and like how expensive the tests are, like with rapid tests versus PCRs and stuff. So there's but applying that to like all parts of your experiments is just a good thing to remember and calculate. So one of the ways that replicates is we have biological replicates, which is basically increasing the sample size to deal with heterogeneity in the population. So in a lab, we can try to get like heterogeneous mouse strains and just more mice. If in people, we can just have more people. So with a biological replicate, it's bigger sample size, which is obviously going to give us more power. Sometimes we do technical replicates too, which is if we have like an assay that isn't it's reliable, but you can mess up when you're pipetting, or like the machine, when it's reading it's more likely to make a mistake or something. Sometimes we do things for example, with ELISAs, we do it in duplicate. Sometimes we'll do it in triplicate, like it shows in this little image. We have 3 samples from the same mouse, and we run all 3 of them to make sure that the assay didn't just get funky, to make sure that they all agree. So you just have to consider expense, time, money, the ethics, like please don't use 15 mice if you only have to use 10, the reliability of the assay, and just, use your judgment, but be aware that replicates are a way to go. So we all know about correlation causation, but I really like this. So put this comic, I really recommend XKCD. So it's a guy, he says, I used to think correlation implied causation. Then I took a statistics class and now I don't. And she says, sounds like the class helped. Maybe. Just, haha. It's funny. Can't tell. So just thought I'd bring up correlation versus causation because they are not the same. For example, this is a really important thing in exploratory studies, which some studies are exploratory. It's cool. They're really useful. I'm doing them. And I'm doing it to inform future research, shape hypotheses even more. But going forward, just be really careful, use caution, modulate your language, say, we found this association in this exploratory study, as opposed to this causes that. We didn't necessarily use proper sampling methods. We didn't control for confounders. We don't actually know what we found, or we know what we found, but we know it's a place to start. And for one thing, if we're doing like a sequencing thing and we're looking at thousands of data points, we have this, if we look at enough things, we'll find association, even if it's not there. So this one is one of the favorites, spurious correlations. Letters in the winning word of the Scripps National Spelling Bee correlates with number of people killed by venomous spiders, which is like probably not a real relationship. But it just shows that if you test enough things, if you test enough of these functions of something correlating with something else, some of them will have a relationship. There's a really funny website. If you Google spurious correlations, there's all sorts of funny ones. But one thing to think about is the biological plausibility, and just like, the results, you definitely always need to consider them as a possibility, but like also don't turn off your brain. Keep an open mind to weird relationships though. And just do further studies before you have claims. One of the things for the really big studies where we're comparing hundreds or thousands or however millions sometimes things to look for correlations, there's a thing called a Bonferroni correction and other analogs, which you may have heard of, just to look at the multiple comparisons problem, making the significance requirements more strict. But still, even with the correction, you just need to be aware when you're interpreting exploratory studies. And this one is really important, that you don't invite Nicolas Cage to your pool party. This is another one of my favorites. So just throwing that in there. So number of people who drowned falling into a pool correlates with Nicolas Cage films per year. And that's just another fun one. Just to really, the exploratory study found something silly. And yeah, so even though like here, like we've heard of p-values, which is like the chance that your finding was due to chance alone, the p-value here is below 0.05, but it's obviously not real. So a fun little thing to illustrate that is this comic. So somebody says jelly beans cause acne and tells a scientist to investigate. They investigate, p is greater than 0.05 probably, because jelly beans probably don't, in this case, they don't cause acne. But if 20 people do it for every different color, maybe somebody says, oh, maybe it's the color of the jelly bean, which I guess it could be like the dye. But for this purpose, we're going to say that they did it for red and yellow and purple and all these colors, but then it was always p greater than 0.05, but it's 1 out of 20. 5% is 1 out of 20. So if there's a less than 5% chance that it's occurred through chance alone, well, then 1 out of 20 times it'll happen. So they find a p is less than 0.05, and then they call the presses. The green jelly beans are linked to acne, and I think it's just a good way to represent the unreliability of this metric. It's good. I mean, it's useful. It's definitely useful, but it's not like gospel. And we also have to remember about sensationalization, that we need to, especially in public health, it's really important to modulate your language. Like make sure if you ever have a study and it has something really relevant and you're talking to the media, like they'll tend to not on purpose, they'll sensationalize. They won't often understand that it's science and things need to be interpreted within context, and they'll really go crazy. And then they tell everybody some fact or association, and then it's changed, because that's how science works. Things, we find associations or potential associations, we look into it, maybe they're there, maybe there aren't. And that's the whole thing we've been talking about with the scientific method. But if we jump too quickly with telling making these claims, it can really, as we were talking before, it can really hurt the public's perception of scientists, which is in the long run quite harmful. So just something to be aware of. And there's all sorts of, so significance hacking is sometimes when you have your data, you can look at it in a bunch of different ways. You can choose to exclude like the outliers, or you can use different statistical analyses. There's always different ways to look at your data, and to be sure that you're not accidentally claiming significance that's not there. You really should describe and write down the way you're going to analyze your data before you analyze it. That's like a rule. You have to do that, because if you analyze it and then you say, oh, well, maybe I'll look at it with this lens, and we shouldn't have put those data points in because they clearly were like outliers. And wait, how about if we use this different test? And then one of them ends up giving you a p is less than 0.05 or some other significance metric, which is sometimes it's done because people are incentivized because they'll get more money, their fundings will get their studies will get funded, they'll have like fame, like it'll matter. Sometimes people they think an effect is there, and they accidentally, just because they're human, like push and don't even realize they're doing it. But it really disrupts the scientific process, as we know. It's really bad for public health, because these are people's lives and health we're talking about. So it's really important. And it's you may have heard of it. I have an article posted about that. People are becoming more aware of it, but there's an article about p-hacking in clinical trials and how incentives shape the distribution of results across phases. This is another one about clinical trials is a pretty scary place for p-hacking and other significance data dredging. That's just something to always stay aware of when you're designing and you're analyzing and you're looking at other people's experiments, is to always be aware. And just quick, as we mentioned before with safety studies, especially in environmental health, like just be really careful when you say something is safe. And as far as operationalizing our variables go, what does safe mean? What do you mean by safe? If you say this skin is exposed to UV light and there's a certain damage that you don't see. Does that mean UV light is in general completely safe for skin? No, it means it doesn't cause a certain thing that you measured. So you have to be really careful and explicit when you're defining what safe means. And when you write your papers or you describe it, you really need to describe the limitations of your study, the operationalization, the limitations of it, the strengths of it, the rationale. And of course, you can't prove a negative. You can't prove that at something there's no association, because that's like a thing. So just to remember that and stay open to new data. Modulate your language, because we're scientists, although that can be a little bit of a problem. Can anybody think of examples where it's hard, right? Because we're scientists and we need to be explicit and meticulous when we present our data. But within the past 2 years, right, it's been a little bit of a problem because for example, with vaccines, if we're saying like, oh my God, like, we're pretty sure they're safe, which, like, guys, like, I'm pretty sure they're safe. But as a scientist, there's not evidence that they are unsafe, except, in general. So, that's been obviously a problem over these past few years. And are there can anyone think of any other, like, what else has happened where scientists, and the thing is, you really do need to modulate your language as a scientist, but, sometimes it does sort of like become a problem if we're too ambitious about saying something safe, right? What else? Yeah. Sorry.
Going along with sensationalism, there's also like pop science, because it's really easy. Like if I'm the anchor on the nightly news, it's really easy to say like, this just in, like especially if it's like a headline that kind of grabs people's attention, and it's not often followed with like, it almost stands at odds with like what we think about with science being like a kind of like a long-standing, sometimes even arduous process. But like everyone wants a quick like sub or like quick title that they can like, that almost has that like shock value. And so you get a lot of like, it's like how science is going to manifest in other like realms, like I said, in the news, or just like more colloquially, is like it can be an issue, like how it's represented and thought of, in the same vein as sensationalism.
Yeah, definitely. That's definitely a thing that we have to grapple with as public health scientists. Yeah.
And Yvonne wrote in the chat about the new Alzheimer's medication, which is quite controversial, and whether it really is effective in treating early onset Alzheimer's, but yet is approved.
Yeah, definitely. Okay, so there we go. We're gonna take a little break. So I'll get maybe 5 or 10 minutes, and then we'll start back up. Stretch your legs or get some food or whatever. And all right, back. So I'm just gonna get started again, and we'll talk about what happens after we do our experiment, our results, just like go over the structure, the like classic structure of a paper, a little bit about peer review, publication bias, and then ultimately interpreting our results in the broader context of public health. Let me share my screen. There we go. Took a little break. Okay. So communicating your findings. What now? We did all this science. We did these experiments. We did all this research. What now? So we write a paper. So just a quick overview of scientific papers, which we're all familiar with, but it doesn't hurt, especially since we're going to be writing one in the class. We'll go more in depth later, but there's a standardized structure. There's a title, abstract, introduction, methods, results, discussion, conclusion, and your references. And it's important to have this standardized structure because these papers are the way that the scientific community sort of communicates, the way the ideas in science and the theories evolve and sort of get refined to get closer to the truth. And it really just like sort of facilitates that and the peer review process. So we're going to do a quick example of like an actual manuscript that we're currently preparing on the epigenetic differences in people with COVID versus not. We're currently going back and forth with our collaborators, and I just want to give that as an example of a paper. So the title, it has the title, and which is pretty descriptive. The aberrant genome-wide DNA methylation profiles of peripheral blood mononuclear cells from patients hospitalized with COVID-19. It's succinct but detailed, and tells, because when you're online or you're looking for papers, you mainly see the title first and you decide whether or not to look at it. So you need to make sure that it's detailed enough and succinct enough and jargon free as much as possible, so that people can get an idea of what's going on. We have the authors, give everybody credit for being on the paper and their associations. So the abstract is just a short paragraph, usually limited in word count, that allows the reader to just like really concisely see what you did, who or what you studied, and just a 1 or 2 sentence conclusion based on your interpretation of your data. And the point of the abstract is, it's like the next step after the title. It's to allow the reader to quickly learn what it is you did, what you found, and decide for themselves whether they actually want to read your paper and then draw their own conclusions, which is important in science too. Read the paper thoroughly so that you can draw the conclusions after looking at their methods and their data. Sometimes, honestly, the abstracts like don't really match the paper, and they're subjective because they're so concise and short. So as a reader, definitely, yeah, it can maybe sometimes be tempting to quote findings just based on reading the abstract, but it isn't a really good idea, because people can be a little fast and loose with their interpretations or their wording, and just sometimes are kind of out of left field. So it's really important to read the paper. I don't know if any of you have ever had that experience when you're doing research where you're reading it, you look at it and you're like, is this an abstract of the paper? Has that ever happened? Do you have any experience with that?
I'll give you an example. One that I read recently, actually reviewed a manuscript like this recently. Tell me what you think. So it was a paper looking at an association between radiation in PM 2.5 samples in a part of the country and a putative health effect in a large cohort of people. And the abstract said we found a very strong association with a very strong p-value between radiation in the particulate matter and in a health outcome, which when I read it, I said, that's kind of a weird association. I can't imagine biologically how radiation would have caused that effect, but their p-value was very clear. So I read the paper very carefully, and I looked at the methods section, and I looked at their analysis of the PM 2.5, and I quickly realized that the actual concentration of radiation in the particulate matter they were measuring was less than 1 count per minute, which means there actually wasn't any radiation in most of the samples they were actually analyzing. But in aggregate, when they were calculating this over the body burden and the amount of material one inspired during a day, they got a value of X number of counts per day. But when you compare that to the actual body burden that we each get from eating bananas and eating foodstuffs and inhaling air and drinking water, each of which has a tiny little bit of radiation in it, compared to our total body burden, it was infinitesimal. It was orders of magnitude less. But when you looked at the statistics, when you looked at in aggregate what they did, they actually got a very significant p-value when they associated A with B. And that's what they reported in their abstract. But you wouldn't get that impression from reading the abstract, that in fact, what they were actually measuring was essentially a non-quantity. So it's important sometimes to read the abstract and understand the abstract, and then go back into the methods of the paper and say, but what did they do? How did they get those numbers? How did they get that p-value? Has anyone else had an experience like that? So in this case, the important take-home message was the actual body burden of radiation from inspiring PM 2.5 was several orders of magnitude actually less than the body burden already in the body of individuals. So clearly we're finding some association, but I don't think they proved that inspiration of radiation from PM 2.5 caused the health effect they were measuring.
Yeah. Right. It's really important to read the methods and the results. Yeah. And to do a good job when you are writing your methods and results so that people can have a good idea of what you're doing. Yeah, so after the abstract, we have our introduction, which has, you know, some background information, just to give context of why we're doing the experiment, operationalizing the variables, and just sort of giving a general framework of the reasons and the background of why you're doing the experiment, because not everyone's an expert and they don't know all of the details. And then you have your methods section, and this is just an example it's the graphical example from our same draft. We have words with detailed descriptions as well, of course, but this is just a nice, easy way to interpret a summary of what we did. And it's so in the methods section, you don't put your data, and you certainly don't interpret your data. You just say what your methods were so that people can make their own decisions about your methods and whether they're reliable, because garbage in, garbage out, that includes methods, and whether or not you did your experiment well, also so that they can potentially replicate it or use your method in a different experiment. One exception to that is sometimes you'll put like a graph or some data that you'll do while you're doing your experiment that validates that the methods were working. So that's not really data that's relevant to the actual experiment, but it's relevant to confirm that the methods went the way that they were supposed to go. And then you have the results section. This is just an example of some of our results. You put the results and the statistics that you use to analyze them. We don't make any interpretations. We're simply presenting the data so the reader can make their own interpretations. A mistake that some authors sometimes make is they interpret the data within the figure caption by telling the reader what they think the data means, rather than simply describing what the graph is and giving the reader a chance to interpret it for themselves, which is the purpose of the results section. And that's why we have the discussion section, is so that we have an opportunity to interpret the data to the best of our ability, talk about the potential limitations, and draw wider conclusions about the generalizability of the findings, and you know, just broadly suggest what the public health implications of the data will be, and anything else we sort of want to say. And then we have a conclusion, just a few short sentences describing the most important findings and interpreting the relevance for public health. And definitely put your references, always cite your sources. But one thing that I would like to convey is that it's our general practice to cite original references rather than citing a reference that like cites another reference. And then you do this sort of like bunny rabbit hop game to find the real reference, and that can get a little annoying for readers. So that's definitely something to keep in mind. And as we mentioned before, graphical abstracts are like so hot right now. They're really they're really popular right now. I think it's probably because like Facebook and stuff made people used to looking at infographics, and I think Elsevier is like requiring them, and I think it's just good skill. So we might work on that in this class as well. And it's very simple. It's just a really quick graphical representation of your abstract, just, you know, who the subjects were, what you did, what the results were. That's it. But it's an important skill, I think, nowadays to develop. So now I'm going to quickly talk about just peer review, which is the sort of quality control metric, quality control sort of process in science, or at least that's what it's supposed to be, and publication bias. So the peer review process is when experts in the field, such as the above, review your study methods and your analyses, and they're very mean. Just kidding. They're usually nice. And so you submit your article to a journal, and if the editor thinks it's like good enough, they submit it to reviewers, and they go through it, and they'll look at your methods. They'll look at your data and the analyses and the conclusions you drew from them, and they'll give you comments about maybe different ways to like make the study more robust, to analyze it. Even sometimes they'll throw a comment back and say, you need to add this whole like extra step in your study, like do more science before resubmitting it because there's a gap. And then eventually after refining it, it'll be like accepted into like the prior in like to like the greater body of knowledge, and or sometimes it just won't be rejected. And sometimes like the as I mentioned before, like the higher journals, the more like Nature and Science and the more they're called high impact journals, they won't be as open to publishing something that's less that isn't as novel or revolutionary. But as I mentioned before, it's really important to publish null results, publish replicated studies. So sometimes you just kind of have to go down the list and go to lower tier journals for that, but it's still important. Unfortunately, there's these things that just to be aware of. They're called predatory journals, where they'll print like anything. They'll print literally anything. You just pay them money. So make sure that you go to a reputable journal with a reputable peer review process, so that you can have something incorporated into the general body of knowledge that should be there. And sometimes it's just lower tier because if you have null results or repeated results, that's just what happens, but that's okay. Null results are okay, and we should definitely still publish them. We do get the publication bias that I mentioned, where people are more excited about exciting results, regardless of how sure we are that they are more likely to be aligning with reality. And you get this thing called the file drawer effect, where there's like a imaginary file drawer where you have your result, you have your experiment, you get your results. Some of them, they're like no results, you don't see a relationship, or it's not really what you thought you'd see, and you just don't even try to publish it because it's not going to be in a high impact journal. But then you end up with an imbalance of what should be happening when everybody publishes their experiments in aggregate. And there are a couple of pretty cool visualizations for this. This is called a funnel plot. So the horizontal axis is the effect size. So in this example, there's no effect, and the y-axis is the size of the study. So the idea is that the higher the sample size, the more precise your results are going to be. So if everybody submitted every single study that they did, just like statistically, it should form a sort of funnel shape. But sometimes if people will do a meta-analysis, and they'll go through a bunch of studies, and they'll make these plots, and sometimes it'll look a little bit like this. Can anyone tell me what they think is going on there? Why there's like a gap instead of a full funnel?
Lack of information, lack of studies published.
Mm-hmm. Yeah, so people looked around in the literature, and they found studies that had interesting results that showed an effect of a certain type of effect. But just statistically, those studies should be there. If in reality the effect size is 0, the other studies should show up on this funnel plot. But since they're not there, it hints to there being like a lopsided presentation of the actual studies studying this phenomenon, which is a problem, because other scientists need to know. And it's okay if it's like a weird result, because that's expected, like this funnel plot shows. But it'll cause other researchers to maybe repeat experiments with a lot of resources that a lot of people have already done, but they haven't published. It's a really kind of important thing to look out for. And nowadays there are more and more meta-analyses and systematic reviews, where people, you can take class on it at Columbia. They go through all the literature. They find things that the variables they're operationalized to be representing the same relationship, and they plot them. And this is called a forest plot. Raised hand. Yeah, what's up?
Yeah, I have a question about the previous slide.
Yeah.
What if the effect size is the true effect size is is positive or above -6. How can we say
of -6? Yeah, so this study is, if the graph is representing like a study that is the real effect size is 0, like this is just a hypothetical graph. But if the real effect size was, like 6 or something, the funnel would just be shifted, right? We'd have the effect size of 0 would just, honestly, it's a little confusing that they did a null example, but if the effect size was 6, you would expect the little triangle to be shifted and be like this. So like it would be the biggest studies are gathering around here because the effect size is 6. And then the smaller, less precise studies would like span out around here. So the idea is that if people were underreporting the results, you'd probably have a gap here because, and again, that would make sense because here you'd see this huge effect size and it'd be all exciting. Here you'd see a smaller one, people might not report it. So it would just shift over the triangle.
Thank you.
Yeah. Yeah, anyone else have any questions or comments about funnel plots? I think they're cool. They're just like a sort of almost qualitative representation just to give us an idea of what's going on. And then, so we have forest plots, which you'll also see in meta-analyses. And they do a similar thing. They look for studies. The bigger little dots are bigger studies, which you can see like the ones with the small dots have bigger error bars. And here, this is, this one is describing like a study that is a little bit like this one is just like a hypothetical one. This one's a real study. Can anyone tell me, like, interpret that graph, what it probably means? So if you saw this, would you think that there's an effect? Like a real one? Or an association, not an effect, an association.
No, I wouldn't say there is an effect because risk ratio in all the 95% confidence intervals include 1.
Mm-hmm.
So,
Well, so one thing about that is that totally, like, there definitely could not be an effect. And if there were more studies, I'd be more willing to think that there was an effect. But if you see, it's sort of like as if this were shifted. If you look really closely, you could sort of see that the smaller studies, right? They do have these error bars, but they tend to be sort of shifted to the left. And if you see that bottom one, it is shifted across the null line, the 1.0. So here, this like the way that one would usually interpret this, but it's like kind of, it's like a 0.7 or something. But it's saying, okay, like we get all these in aggregate, and yes, the error bars are here, but the whole point of doing all these studies is to look at them together. And if there's like an appreciable trend in one direction, this makes us think, oh, like I think there's probably an association here. It gives you a way to contextualize all of these things all together, just in like a graphical form. If you like, because like if you see over like here, there's one.
Oh, yes.
Can you see my mouse?
Yes.
Yeah. Here there's like one that doesn't hit that, but on this side, there isn't a single one that has like a main thing without crossing the error bars over here. I mean, that's only one, but it's just like the general trend is on this side.
Okay.
Right. So that's just another, it's just a cool tool to see, look at meta-analyses and help you with your research, trying to decide, whether there's like, what hypothesis you want to like look at and what you would expect to happen and what's worthwhile and what to use for making recommendations. Yeah. So yeah, after publication, your results are available. Now they're in the greater scientific body of knowledge. Yay, you can help improve and modify the theories, get closer to the truth. Other people can do your experiment with similar methods or the same ones, and you can inform your future hypotheses. So, and you might be in a meta-analysis. So what comes next? You go through, you look at all, see, there's another one. This is a better view of a forest plot, having like a pretty clear looking effect. Like this looks like there's a pretty reliable effect, even though a lot of these error bars are here. Like we're not 100% sure, but the trend is definitely over here. And the aggregated one is like this. Yeah, cool. All right, thumbs up. Yeah, so we've done this. Now we look at all of the evidence together and we start trying to apply that to public health to make some recommendations, which was, the science is obviously really important. We need to, yay, we did it. It's really important. And now we take that, and as public health scientists, we take the refined, getting closer and closer to the truth, and try to start applying that to public health. So again, as with the scientific method, the traditional one, you have your cycle, everybody comes together, you refine the truth over and over and over again. And it takes a lot longer for people to say, oh, I think that there's an effect here. People in generally, in like maybe like a physical science, will be more hesitant to like claim something. But the thing is that in public health, we need to know, we need to make a recommendation right now. So we need to just use the data that we have and the studies that we have and the research that we have to make the best recommendation that we can. We'd like it to be validated by like hundreds of studies, but right now we need to know what to do, what guidelines to set, what recommendations to make. So it has like a slightly different sort of flow to it, even though it is a scientific method, but it sort of gets chopped up every once in a while because it's the same idea, but sometimes you need to make a recommendation. So it's the same as the scientific method. You wonder why something's happening, come up with a reasonable testable explanation, operationalize your variables, make an experiment, do an observational study, get some data, look at what it means, decide, the most likely interpretation, and make the best recommendation you can. Constantly look for new information all the time, same as the normal scientific method. And then something else might happen. Some study might come out, some really clear trend might sort of like emerge, and you have to so you do it again. It's a scientific method. You keep going over and over, something new happens. So you update your explanation. You look at that new data, combine it with all of the old data, get like your best updated view as possible, and do your best to make the best updated recommendations with the current data. Something happens again, you get more data, same thing, update your explanation, look at the data, get a new view, make new recommendations, and on and on and on. That's what happens in public health. So it's the scientific method with like little stops where you have to like force yourself to make some decision the best you can. So can anybody, there are pretty obvious examples of that happening right now. Can anybody think of any particularly salient ones within the past 2 years? Times when we had, we made recommendations, or the people made recommendations and then they got new information and then had to like update their recommendations.
So think of March, April 2020. Think of Anthony Fauci and the CDC and some very early there you go. Exactly right.
Yes. Mask wearing. Yep, exactly. That's like the most infamous one, right? Trying to balance the data that's available, which is like basically not much because it's a novel virus, with the resources available and everything. And I think we've all learned a few lessons. Isolation periods, absolutely, right? We changed it from 14 days to 10 to dealing with what we think people will actually do. Yeah, absolutely. This has been a very obvious crash course in public health for the entire world.
Fomites, another example, right? The ability of this virus to be transmitted from your UPS parcels compared to aerosolization.
Right.
We were all leaving our groceries out for 2 days before we ate them.
Oh boy. Yeah. All right. So, go ahead.
It's a little off topic. It's not particularly salient.
Oh, no, I love off topic.
We used to think that pandas wanted to live in isolation and they needed to be alone. And it turns out they're very social animals. So all these years we've been putting them in their own separate pens, and they haven't been, mating. They haven't, and then it's been sort of a new development. They're actually very, very social. So it's likely they were lonely.
Oh, that's so sad.
Yeah.
Little pandas.
Offer topic.
No, that's not off topic at all. This is yeah, that's exactly what we're talking about, right? We thought we were doing the right thing, and we just had to really, keep looking for new information and stay open. And that's another important thing. Really stay open to new information. Like people might say, "Oh, but I think the pandas are lonely." And they go, "Come on, everyone knows that pandas they're not social and they want to be alone." Well, apparently that wasn't true, right? So that's why it's so important to keep an open mind. And update I love that. Perfect. Yes. Maybe it's best to not talk about COVID all the time. It's so depressing. Thank you. Yeah, exactly. Yeah. Yeah, that's it. So you guys are becoming part of the scientific community. Welcome. We're going to learn some skills, and combining them with the scientific method and everything we're learning, you'll be able to contribute meaningful work, critically interpret others' work, and communicate that to the world at large, which is especially important nowadays. And chat. Oh, you're welcome. And that's the end. So.
Any comments?
Hey.
Conclusions? Well done. Wow, lots of hand
Oh,
clapping.
yay. Thank you.
Good. A very brief tour into science.
Yeah.
Good. So we'll post this lecture, and we will post some of the files and documents into CourseWorks and also into our Google Drive. Everyone knows the address, right? You sent that out.
I'm going to send it out again.
Okay.
And I might I just might type everybody's unis at cumc.columbia.edu and just invite everyone individually. But we'll definitely, I'll just make it a practice to, whenever I post something, I'll post it in the files tab on CourseWorks, and I'll post it in there. But the collaborative documents will be in the Google Drive. So
good. And we will see you all next week in person, God willing. And we will start off in the conference room on the 16th floor. We'll give you the room number for that. We'll have a brief discussion about science of the day or something news we might have read about. We'll have a brief discussion of some of the formulas and approaches, things like just making a concentration or percent or molarity, just so you have a brief overview, how to weigh something, how to pipette something, couple of quick demonstrations of that. And then we will get right into starting with some sample collection. And for those of you who have volunteered to help with sample collection, we will take some blood and saliva and some nasal swabs, and we will absolutely pick our main questions for the semester during that discussion. But I want you to keep thinking about that during the week and think about what other questions can we be asking? What are the thematic things that we can be doing during this semester that we can use the tools at our disposal to answer or to try to answer? And it'd be really good if we got some real world things that might have some environmental health and public health impact, rather than simply learning some basic technique that doesn't have any direct application to things that we're passionate about or interested in.
And that there's on the Google Doc, there's everybody has access to edit, and there's just a document. So just throw your thoughts in there. A couple of you guys I saw put some stuff in, so yay. If you have any thoughts that we hadn't brought up, just throw them in there. Put any, thoughts about it, experiments you can think of, maybe background research, whatever, just throw it in that document, and we can all use it to sort of collaborate and stuff. Yeah.
Excellent.
Yeah.
Any other comments? Feel free to email us at any time, call us. You have our contact information, and we'll try to answer them. All right.
Thanks so much.
All right, everyone have a great week. We will hopefully see you all in person next week. Anay and Lauren, if I could ask you to just hang on for a minute and talk about some lab related stuff, and rest of you, we'll see you all next week. Stay safe.